Hey Mustangs, in this video we're going to be taking a look at the experiments that were used to identify the structure and function of DNA. So, nowadays it seems really obvious to us that DNA is the genetic material that gets passed on in order to cause disease, in order to transmit traits from parents to offspring, but it wasn't always so obvious, and there were brilliant minds that actually worked on this question of what is the genetic material? What is it that actually gets passed on? And there were three main ideas. Some scientists thought it was protein, others RNA, and others DNA. The two biggest ones were actually proteins, and so if you take a look, here we have four major experiments. We have the Griffiths Experiment, the Hershey Chase Experiment, Char Goff's experiments, and Rosalind Franklin's X-ray diffraction. So the first one, Griffiths Experiments, this showed us that DNA could transform bacteria. So that DNA carried some sort of instructions that can change a bacteria that was not deadly into bacteria that was deadly. So how did we figure this out? Well, in Griffith's experiments, he took bacteria that was deadly. If you see this pink kind of outside that represents the deadly bacteria, he injected it into mice and it killed the mice, as expected. He then took bacteria that was not deadly, injected it into mice. The mice were perfectly fine, as expected. Then he killed the bacteria that were deadly, and when he injected them into mice, the mice were perfectly fine. So something in here, something in these bacteria basically was destroyed, and as a result of that, it did not make the mice sick. So what he did was he mixed bacteria that was deadly but had been killed with bacteria that was not deadly at all, it had never been deadly, and injected it into mice. Now you would expect, okay, if we look here, the heat-killed ones, mice are fine. The non-deadly, mice are fine. So you would expect that the mice would be okay. But what happened was the mice died. And when he took samples, he found deadly bacteria. So what happened here was these bacteria that were once the deadly kind that had been killed, passed on DNA to these non-deadly ones. So these non-deadly bacteria picked up DNA. We call that transformation. Whenever bacteria do that, they can actually pick up DNA from their environment. So since these had died, these non-deadly bacteria picked up the deadly instructions in the DNA from these guys, and it transformed them. So that's why we call it transformation. Okay, so these became deadly bacteria because they had the instructions to cause them to be deadly, and it killed the mice. So this showed that DNA obviously transmitted instructions for the bacteria to actually become deadly. A big one here is the Hershey Chase experiment. So this one is huge. The Hershey Chase experiment kind of broke down the debate between whether protein was the information that gets passed on or if it was the DNA. So key thing here is to realize that in this experiment, it involves viruses and bacteria, and then radio-labeling techniques. Okay? So viruses can infect bacteria. A lot of people don't think that bacteria can get sick, but they actually can. So viruses can actually infect bacteria, and viruses pass on certain instructions that cause the bacteria to make more viruses for the virus. So in order to find out is it DNA or RNA, these guys are really good to use because viruses are simply DNA, usually, could be RNA, but it's usually DNA, with a protein coat on the outside. So you have the two things that scientists were looking at, and obviously whatever gets injected in the bacteria must be the thing that is passing on instructions to infect the bacteria. So it's either the protein or the DNA. And we use radio labeling here. Remember, radio labeling is kind of like a tracking device. When a molecule becomes radio labeled or an atom becomes radio labeled, it's really easy to track it and follow it through an experiment. So we use radio labeling a lot if we want to track molecules and find out where they are. So here, one batch of viruses was radio labeled with their protein. So here the proteins get labeled, and it's the sulfur in their proteins. And what we see here in pink, that represents the radio labeling of the protein. So that becomes radio labeled, if that's what gets passed on to the bacteria that caused the bacteria to become infected, then that should wind up inside the bacteria. So they radio-labeled it, so we see it in pink, radio-labeled. Here's the bacteria. They put this in the machine to separate out the viruses from the bacteria, put it in a centrifuge. The centrifuge spins, and the bacteria are heavier than the parts of the virus that are left. So at the end of centrifuging, at the very bottom, the bacteria sink because they're heavier, and there was no radio-labeled protein found in the bacteria down here. All the radio-labeled protein was found up here in the viruses where the bacteria was not. Okay. Another part of this experiment was radio-labeling the DNA. So the phosphorus of the DNA was radio-labeled. So here we see it in blue this time. Now we see gray on the outside to represent the protein part of the virus. So here we have the viruses, here we have the bacteria. Do the same thing, so same model that was used before. Let the viruses infect the bacteria, put them in this machine to separate the virus parts from whatever got injected into the bacteria here, and then centrifuge it. And if DNA is the material that got passed into the bacteria, then we should find it in the bacteria. So bacteria, once again, they're heavier, so they sink to the bottom. The parts of the virus would be on top. And lo and behold, we found radio-labeled DNA inside the bacteria. So that showed us that the DNA was passed on, not the protein. All right, Char Goff's experiments basically allowed us to see that A and T pair together and G and C pair together. So if you look, you find ratios of different ones. So if you find that in a certain DNA molecule, 40% of it is T's, 40% of it is A's. So the ratios match. So they go with each other here. So you will always see that A and T will always pair together, and there will be equal amounts of A's and T's, and G and C will always pair together, and they will always have equal amounts as a result. So Char Goff's experiments basically help identify the base pairing rules. And then finally, DNA structure. We know that DNA is a double helix, anti-parallel double helix. It has a 5' end, 3' end, and then the other strand is reversed here. And we know that there's two strands held together by hydrogen bonds in the middle. The covalent bonds hold these here. But how did we figure this out? And it was Rosalind Franklin's X-ray diffraction. So this picture of a DNA molecule, this X-ray picture, was basically used to find out that DNA is a double helix. So this simple X shape that you see doesn't seem like much, but that shows that DNA is a double helix. And we're also able to calculate the size of the DNA molecule, how wide it is, and also how far apart the bases are.